I'm not seeing that salty halo.
I think the
distortion
angle
at
on
the
here, I'm
top.
not squeezing as hard. I'm not flattening the tube as much as I was on the other ones. So.
Yeah, so I think we're probably going to do a fourth squeeze. This is the third, correct?
Yeah, that's the third.
Correct. Okay, ready for 4?
Sure, if you're...
There we go.
Yep. Now just give it a second to... Oops.
Oh,
Okay.
there's
That did
a
it.
lot of stuff.
So, you want a 4th or you good?
I guess that's good. I hope it's seated. So let's seat it back because it looked like it had kind of come off.
wide.
So hopefully the core is still in place.
I'm still seeing a lot of that distortion around the flap. So it's
Well, yeah,
some.
like I say, hopefully the core is still where it's supposed to be. So
yeah.
Sure, go ahead and seat that again.
Seating.
But you know, to tell you the truth, I would really like to squeeze that one once more, if
Me
we
too.
could. Oh, awesome.
How about a fingertip squeeze?
Sure, try that. We'll see what happens.
Seeing flow.
Out the flapper?
No, in.
No, in. Okay.
Yeah,
I see. Okay.
There's some on the side as well.
I see.
see outflow now. Top left.
Okay. All right. So I think we're good.
Yeah, there's a lot of outflow now.
Okay.
So let go.
Yes, please.
Let go.
Okay.
Might be the best one yet.
You want to reseat or are you good?
Yes, just go ahead and try to reseat it. And also, well, I think you did the other ones also. Just a little.
Yeah, give them a little love tap.
Yes.
1.
Okay.
All right. Now we have to do the hard ones. Well, no,
2.
actually, these were the hardest ones for today. The other ones we just put in the seafloor. They can't.
And leave them there for...
Yeah, we put them in, we inject them, and we leave them. And we get the lights off of them as soon as possible. So they're all in the on the starboard side, which I know is not the favorite location.
Oh, it's good for us.
You know what? I'm going to say, can we shoot 3 or 4 Niskins? How many Niskins do you have? 3?
6.
Yeah, well, let's shoot 4 of them.
Right here?
Sure.
Right now?
Yeah, it's pretty clear. So let's do that. And then, can we do, let me check my watch. Let's do a 10-minute par, all the lights off.
See this? Oh, yeah.
So can we do that? I have to
Sure.
Step out for a moment. Yep.
I'm going to step out while we're doing that too.
I don't know what's up with this.
That thing makes me dizzy.
Why is it not angled right?
I don't know, but I don't mind looking at the jewelry on the front there to make sure I'm not kissing it.
Am I back in?
I don't need it there.
Yeah,
you can
so.
put it back where it was. I know you like moving it around to make me dizzy.
Where's
when it's looking down like that, I can tell if I'm too high with the manipulator. So I can see it before I hit it, kind of thing.
Where's your bio box cam? It's off right now?
The what?
The starboard bio box cam.
It is not on.
Oh, no, I see it now. It's that fisheye.
Yeah, that's one of them, yeah.
So that
I
needs
don't know
to
where
be
the
on.
other one. Oh, I have to switch my
You have
cameras.
to switch it to port. I was wondering why I couldn't.
Yeah, I do.
Yep. That's one of the shared
Okay.
Right,
connections coming up.
Are we ready for Niskin number 1?
No, we want to see
Hold
it.
on,
Oh, yeah.
hold on. I got it. I'm on it.
You got it. Get it. Get it.
Hotel cameras. See, I can sit over there.
Okay.
Let me stream it. Stand by. Oh,
Roger.
there we go. Looking at some Niskins on sat feed 3.
All the way in the back.
Anyone want to call it there?
Stand by. Let me do it on my monitor. I got a great monitor. All right, I'll call it.
Say when.
I haven't seen it. They're still all cocked.
Look at you, multi-viewer go.
Yeah.
Yeah. What one was that?
That,
Multi-viewer.
that's no, this is my PIP. This is one of my.
But how do you switch it into that one?
I just went to PIP and then switched it over on my
Yeah,
port bio.
PIP. What is it? PIP 1?
Yeah, for me it's PIP 1.
Roger.
Yeah, it's H1.
Okay. So
PIP 1.
am I pulling this thing or what? Are you ready?
Yeah, I'm ready.
I'm ready. Science ready?
Yeah,
Yep.
we're ready. Another back. Oh, Niskin triggered.
Did it
All
go?
right.
Did it
Yep.
go?
Yeah.
Yeah, one's
So
in the
all
back.
the way in
Yep.
the back, yeah.
So
It's kind of
They're
backwards.
just looking on the camera to see if the Niskin triggered.
Okay, number 2 now.
They pull it and then it closes in the back, and you can see on that lower camera, yeah. Yeah, so they're back to front.
Ooh, can I do 2 at once?
Don't.
what are we doing? We're doing more. Oh,
Or-
Niskin triggered.
Okay.
Niskin 2 triggered. Ah, get off me. Number 3, right?
Give me 1 second to catch up.
Roger.
Number 3's got a colorful hat.
Okay, you're doing 3 now?
Copy, this will be number 3.
Okay.
Ready?
Yep.
Here we go.
Got it.
Niskin triggered.
And you want one more, yeah?
You wanted 4 total, right, Jen?
Yes, please.
Niskin
Got it.
triggered.
That's number 4.
Okay, now could we turn off all the lights and
Yeah, let me
mark the...
Let me get out of here with
right.
Snaggletooth here
Roger
without
that, roger that.
disrupting
So
any
Just,
jewelry on the front.
sure. Just obviously when we do it, we want to set the time because we need to
Yeah.
know for the PAR when the
Absolutely.
lights are off. Yeah. And all of the lights are masked. I mean, those LED lights
Yeah.
on the...
We have one camera that has LED lights that we need to turn off.
that would be nice.
That would be.
Imperative, please do so.
You want to retract the porch there, Michael?
Yeah.
I think we're done with that thing.
I believe you.
All the way in. Is that it?
Yeah.
Yeah. Okay. What are we doing? Lights.
Turning up all the lights.
Alexa, ROV lights off, please.
Yes. You know, like half of our listeners are now listening to the smart speakers say, I don't know that command.
And that
Argus
camera,
Argus 2.
that camera should be off.
Yes,
'cause
Argus also, please.
I'm seeing image on,
there.
you go.
So that's bucket cam, right?
Yeah.
See in the bubble cam. So that bucket cam has LEDs on it. So okay, I still see lights. What am I missing?
I'm not seeing Any detectable. What do you see
Okay.
lights?
Power sensor. That should have gone from.
All right.
Oh wow!
So starting that now.
It went from a luminance of decimal 0.
And how many minutes did you want to do this one?
Let's do
You
10.
see it going
10.
down.
Yeah.
I'm going to increase the gain on the Zeus camera just a little to look for some dead pixels.
It's funny, it
1,
didn't change a lot.
2, 3. Dan, you see 3 dead pixels
What's that?
on the Zeus?
Oh,
you
yeah.
see the 3 dead pixels?
Yeah.
Wow. Should take a video still of that.
You want it framed for your wall?
What is that movie?
Poltergeist.
Poltergeist. Poltergeist in color. This
I
is
have no
Poltergeist
idea what you're
remake.
talking about. What movie?
So our luminance went from decimal 0. Our power luminance went from decimal 0106 to decimal 010. Sorry. It was between 7 and 8 and it went to 6.
Yeah, I see that on your screen. I'll write that down.
You got it up on the
Yeah.
graphometer. So 10 minutes of this?
10 minutes. So that'll be 11:04.
Yeah, right, if I do a bio break?
Go for it.
Oh, thank you. Mike, you all right?
Yeah.
I'll be back in 5 minutes.
All right. I'm going to turn my gain back down. That's 42 dB of gain. I even have more, I think.
What?
48. Yeah, 48 dB of gain.
I want to
That's
touch it.
18.
Don't touch it.
All right, so for those of you viewing, dark that you see on SAT-1 and SAT-2 are actual ROV cameras. This is what the ocean looks like at night to a video camera, or during the day. When you're out of the photo
knows?
zone, down below 200-ish meters.
Yeah.
So just for people that might not have been following along for the past couple of days, we have this one species of foram that sequesters chloroplasts from diatoms. So of course, chloroplasts are the photosynthetic organelles, and they capture energy from sunlight, and that's how plants basically make their living, or how they survive. So, and these forams they look green. They're living down here at about 570 meters, 571. And actually they live down to 595 meters, which is the deepest part of the basin we've sampled I've sampled over the past 30 years. And so the question is, what are they doing? Are they just getting sunlight where they're living? And so we're doing these power sensor episodes so that we can document how much sunlight is down here where they're living. I mean, it's kind of like, it might seem a little silly, but we have to do it. So that's why we're doing this.
Joan, so what, you guys tested those squeeze scores last night. What did you guys do? Use dye or something to see how they work?
So last night, well, earlier this morning, the dive, I think it was from 12 to 3:30 or what have you.
Roger.
I have to admit, I actually did go sleep for about 2 hours. So I was not there, but we filled one of those large squeeze tubes that we just saw. We filled it with just water and red food color. And we just wanted to, well, we were doing a few things. We wanted to see how well the cores would work and the T-handles would work, because there were some discussion from the prior dive that just only had the one kind of grip, at the top of the push core. So that we wanted to deploy a T-handle. So that was one reason. And then we also wanted to see how the squeeze worked. And so today we just did it on 3 cores, but we had an actual chemical in there, and it's a very high salt chemical. So it is much denser than seawater, And that's why we could kind of do it this way, where we could have 3 squeezes, because the seawater that came in from the top after each squeeze would stay on the surface because the chemical that we were injecting was denser. It just stays at the bottom. But and so this is basically a way to preserve the cells on the seafloor, so they never have seen sunlight. So they've seen a little bit of ROV light, so granted that is the case, but effectively they haven't seen light.
So I never saw it correlate that as a pretty common coring.
I've done it on, it's not common, but I have done it on way back in the, in the early 2000s with Ambari's old ROV. I think that's where I got the idea from them. And then I've done it with ROV Jason in the deep hypersaline anoxic brine basins in the deep Mediterranean off Greece. We actually had a dive and discover episode for that. So if you ever know what dive and discover is through Woods Hole Oceanographic, we have an expedition that covered that whole effort. That was, gosh, I think it was about 10 years ago now. And when else have I used them? Well, those are the ones that come to mind right at the moment.
Very cool.
We've never done ones this big before where we do these multi-squeezes. So this was a bit of a tweak, and it was a little bit experimental, but I work with some very smart Alvin Jason engineers at HUI. Well, one in particular who happens to be my husband. So we've spent a few weekends working on this.
Nice.
Yeah, you know, things that they never get done 9 to 5 weekdays. So I have to say, Phil, looks like they worked.
Excellent. We have some viewers wondering things about the sediment, like how thick the bacterial mat is and why it's so dark at the bottoms of the cores.
Right. So the bacterial mat, as you might have seen, the thickness depends on exactly where we are at a particular time. It looks like here it's maybe a couple millimeters thick. It really is like the white fluff, if you want to call it. But sometimes it also is kind of, as I was saying the other day, a felt, kind of a mat of felt. And that's why they're a little bit cohesive when you push the push cores into the sediment. And the reason everything's so dark is because the oxygen goes away here. We've been measuring it with microelectrode profiles. It goes away within the top, certainly within the top ½ centimeter, probably less. We think that we're getting a little bit of oxygen in the surface waters when we're taking them out of the quivers. So, but you know, the reason they're black or dark is because there's no oxygen. And they're very high organics. This whole area is very productive. So you have a lot of organic material, as you may have noticed, coming through the water column for, you know, ascending or descending with the vehicle. So you can see all that marine snow and then all this zooplankton that eats it, and ultimately the zooplankton will make fecal pellets, and those settle down to the seafloor, and of course sometimes phytoplankton die and will settle to the seafloor. So it's very organic rich. That also adds to the dark color.
Awesome. Thank you for that description, Jen. Well, it looks like we're coming up in just about a minute on 10 minutes. So
Are you calling us?
I think, are you talking to me, Dan?
No, sorry. Talking on the bridge. Video stepped out for a second. So did now.
All right. So I'll note how long it is when they get back.
Okay.
Yeah, I'll let you know when he gets back.
Right. So just to let you guys know what we have to do, what more is to be done on this dive. We need to take those 5 push cores on the starboard side, or the right side, as you, whichever.
Whichever.
Yeah. And we need to place them into the seafloor and leave them there, inject. We only do one squeeze. These don't have check valves to let seawater in. So we would like to focus in really closely. Hopefully we'll be able to see the injector, the end of the capillary tubing, where we should be able to see some very cloudy, it's really more of a slurry, but it's a liquid. We should be able to see that come into the core overlying water.
Where are we on the power timing there?
We're done, so whenever Michael comes back, I'll note that time.
Or just they have to start turning lights on and stuff.
Yeah.
So we're done with the 10 minutes, so I can turn on lights?
Yep, I'll note it.
Roger. Computer, lights.
Smart speaker.
Action. I think we should just stay here because we've had success with these squeezer cores, and of course,
Can.
so those are relatively heavy compared to, of course, the other ones, which are a little lighter. So hopefully these will stay put. And we're putting them down pretty far, so hopefully, hopefully it will work.
But we'll want to be able to see that top capillary tube coming in, right? So
Yeah, I'm not exactly sure the way it's going to,
yeah.
but I'm rather confident with these.
Great.
That they're not going to blow out. Hopefully we can see some evidence. I didn't want to put food color in this because it's a different kind of chemical that I didn't know what that would do.
Okay.
Chemistry-wise.
When we were doing these squeezes here, the way it looked coming out looked like when you work in a brine pool, you see those kind of,
well, that's
they look clean, like you're working the med.
And that's the point, And so it basically pickles them in place, if you will.
Yeah.
Yep. And so these are the ones we just did, the large squeezers that I'm going to be spending hours picking under the microscope as soon as we get them up on the surface. So
Did you bring Visine?
Oh, I have many different kinds of eye drops. 2 of them prescription. Literally. That's because I have dry eyes, but that's different.
Yeah.
Anyway.
And the forced air.
And of course.
Vessels.
And no sleep.
Yeah. That was exactly where I was going.
Haven't really had much time to eat, but that's not so bad. And we're waiting for the...
Michael's coming back.
Okay.
All right. Are we ready?
And we have like a half an hour.
Yep.
Yeah. So it's 11:08 now.
Time's tight.
Back on SPL. Good.
Yep. Time to go.
Yeah, we've got about a half an hour. So if these go well, it'll be great. And they'll go quick, but we know things don't always happen the way we want them to the first time.
Are we able to get the dash cam, Argus dash cam on this big one instead of the winch?
What salvo did you just fire?
I didn't, nothing.
Huh. It just went up there?
I just sat down and it was there.
Oh. Yeah, that's weird. So on ROV1, well, I wonder what the last salvo you guys fired was. Should be source ROV. It's Argus.
I would be very happy if we get 4 of these. So if you want to start with the easier ones first, but you can start with the harder ones. That's fine with me. Awesome.
Yeah, same game here. If I do the ones in the back, then I won't disturb.
Okay.
They'll be happy.
And then I'm going to go ahead, since we're working over on this starboard side of the vehicle, put that image over on our sat feed 3.
And just remember, we don't want to squeeze the squeezer until we're in the seafloor.
Copy.
So these are going in, getting squeezed and being left, right?
Correct. And so we obviously need to get back here.
Yeah,
Which will be tomorrow night is what which is going to be pretty late. But
Yep.
yeah, well, anyway.
I'm pretty confident
Yeah.
we've all our hits are within a meter and a half.
No,
so.
I'm talking about in terms of getting off the finishing.
Oh yeah.
Okay. There's that little issue.
Well, as long as the time allows, we can get back.
Yeah. We got to figure that out. There's the issue of the abyss that did not get deployed this morning because of some electrical issue, and we're working on that.
Roger.
Technical difficulties, please stand by.
So for those of you watching the website channel 1, all the action's over on the starboard side right now as we work around our bio box and additional Niskins, and that can be seen on channel 3.
So you want to bring these around into the Zeus, obviously, so we can...
Yes, I don't know what the Zeus is exactly.
Sorry,
but
our main HD camera.
Roger that. I think where we were coring was clearly successful, and so hopefully we can carry on that.
Duck is still out there. Like he just swam by the... There he goes.
I was going to say a duck on the seafloor.
I just looked
that.
at the down Argus thing, and he just keeps swimming by.
Oh, yeah, I saw him this morning when you launched.
He's very persistent.
It's the same one.
I think so.
Okay.
So placement.
What's it doing all the way out here?
Oh, right. So,
We're not that far out.
so let's see, can we zoom in to the core top, around those black lines right now so we can get some orientation?
Ready when you are, Dan.
Yeah, let me put it somewhere where you can
that must be the rubber band that's
that looks
hanging
good.
down.
Let me do a partial and you can
Yeah,
align.
it's a rubber band.
Roger.
Yeah. Well, that's going to be
Down in
annoying
here
because
somewhere.
I'm looking for the end of the tubing.
give me
a
So,
Yeah.
can you zoom in there even more, please?
Yep.
I'm looking for...
On the tubing itself?
Yeah, so I can explain that the tubing, I think, would be if you... I don't know that you can do this, but if you rotated to, let's see, counterclockwise, maybe 90 degrees. Yeah, a little more. So we might be able to see it. Yeah. Can you see it under that second hose clamp? It's a little out of focus. There we go.
Yeah, I can see it. The
So
tube that's going
if we could go in this way, and obviously come out this way, and then we do the. And I think that this works well because I assume that you're going to want to squeeze kind of on this side. So.
Yes.
Okay, that'd be great.
Okay.
And now on this one, because we want the volume, so remember the other one we were coming in below the black line because of the quiver, the bung. Here we want it between the black lines on the seafloor because there's no bung, bung during the injection, obviously.
A little wider.
See where we wind up here. I can adjust the camera again. Should be good for some zoom there. Tilt down just there.
Thank you.
Okay, and zoom in a bit.
Come in. Turn around there. Then
Yeah,
I'll give
beauty.
you more once you get in. Well, we'll get the wrist set up.
Okay, y'all ready?
Hold on, before you go in, Dan, there's an object on the seafloor rolling in from the left. It's behind that bent tube right now.
Huh.
It's either, I don't know if it's going to come your way or not. You should see it in just a second. I just want to make sure that's not going to interfere with your operation.
Oh, yeah.
Oh, I see. Uh-huh.
Just let go.
Talking about dead zooplankton.
Yeah.
Freaking passerbys.
Just didn't want it in our sample.
Yeah, I do not want that.
I think we're good there.
Sure. Thank you for that.
Yeah, nice catch, Ed.
Okay. Good luck.
Here we go.
You can't see the surface.
Yeah.
Yeah, it's going to be hard to tell. It's very gelatinous.
Yeah, sometimes they punch and stay. What do you want? You want to abandon and try again?
I don't think this one will work. So I don't know why it's soupier now than,
I don't know either.
45 minutes ago.
Yeah, it went way down.
So in this case, we want to get rid of that because,
Roger.
of course, it's probably going to be persistent and stay. Now when we want to get rid of it.
So
Just-
why wouldn't we like just continue to push it down a bunch with somewhere else?
She wants to be able to see it.
Yeah.
You want it between those two lines. And you can't
Okay,
see the
and the objective is you'll notice that the top of these push cores is clear. It's transparent, and they're going to stay on the seafloor for, well, 24 hours or whatever it turns out to be.
Getting ambient light.
Trying to get the ambient light that may or may not penetrate. We're trying to document, quote unquote photosynthesis. So yeah, if you could get that a little clearer, that would be fine. It's not imperative that it be absolutely pristine. So, yeah, so I'm not sure if we have to go in slower, faster. I don't know, but you had it down before.
More to the right where that other one was.
Yeah, maybe the gray stuff is a little.
Any of these places, I'm fine with. Whatever works for you. And
I
again,
know it's not optimal for camera, but that's good there.
Well, I think we would be able to see.
I will say for recovery, if we can get them in a sequential line, that'll be easiest for everyone to keep track of which are which.
In that case, I would probably move forward a bit. It's going to be hard to sequential line.
All I can say is we have less than a half an hour.
Yeah.
So I don't know.
Okay. We'll try and log it well, whatever ends up working then.
You want to do a quick hop, Dan, and I'll run around the boat resetting the clocks.
Yeah, because it depends on how close I can get them. I mean, we can go out here.
Actually, I think you could get them close enough.
You just won't be able to zoom in because that's all the pan, right, that we have.
This camera can't turn. Yeah,
I see.
So can't turn anymore.
I could turn the vehicle heading a little.
Yeah, that's what I was thinking. Is just do a float up and turn to starboard.
Up to you, Joe. I don't know. I mean, these could go really quickly if we get set up right.
Yeah.
So.
Well, if you want to be able to zoom in on the the things, then we need to move.
Go ahead and do what you need to do. But I don't want to totally relocate because this was a good spot a little while ago.
I'm just going to move a little bit here.
While we're moving, is it, short answer, understood that these organisms are photo or chemosynthetic?
They have, They have all the genetic machinery to be photosynthetic.
Okay.
But they're living down here.
Yeah.
And it's I call this
yeah.
the chloroplast conundrum.
Right.
And they're the hosts of these chloroplasts are very, very abundant.
Got a little brighter on.
That was the light bar.
What light bar?
The one that's a little more rounded off now.
Yeah, we're polishing it. When I zoom in, Dan, I'm not going to be able to view that stopper part of that one that's sticking way up there to the right. I think we're done with it. So
What's that, Ed?
when I zoom in on these guys, I'm not going to be able to keep an eye on this thing. So you have to watch.
Yeah, we don't
As
need
you-
to for
Well,
these.
as the arm comes down, I just don't want to.
I think I'm clear of it.
Okay.
Oh, I see.
Why don't we put bubble on it to
Yeah.
show me the jewelry there in the for the treasures there in the front. Down, down. Yeah, that's what I want to see. Okay, you want to zoom in there a bit, see how we go?
Yeah. Something like that. We're really focused down there.
Okay, ready?
And you want to place it right where we can pick up without it then getting disturbed.
Yeah.
Pick up the vehicle.
Yeah. Well,
Happy here?
Yes. I think it'll be okay. Yes.
Here we go. Say when.
Go.
Maybe slower.
That one seems to fail. Pull it out.
Yeah.
Now,
You know what? Just go, well. I was going to say we can build a stack.
yeah,
Almost.
I asked that earlier, like, but I think my question got, like, what's wrong with just kind of stacking it
Well, for this
And
actually,
until you get it
I
where you want it?
actually don't think that's a problem, to tell you the truth, because we're only going to sample the surface. So
Oh
in theory, actually, we could do that.
do the plunger action.
Yeah, you put it in,
stacking.
and if it doesn't go all the way, take it out, move over a little bit, and dunk it again. So
Yeah.
you just start,
right.
you're just stacking cores,
Exactly.
essentially.
I know it's a little weird, but it's a way to
Right.
get the headspace correct.
You're using the additional layers
And
as
we're-
packing.
And we're exactly, and we're
Okay.
only going to sample the top couple centimeters.
Yeah.
So it doesn't matter what's kind of underneath it.
Okay. So I got a funky grab. I can't reach out too far with this one. I'm gonna go here on
Yeah,
this.
see that? You have your wrist straight down, right?
Well, I had to get on the thing.
Yeah.
It's that far one, so it's okay. You can zoom in there.
I think that should be good.
Here we go.
How far off bottom you are. Oh, I can see now.
I think I should punch through slow and then go fast. So you want to try the stacky thing?
Yeah, I
Yeah.
think so. Let's just see what happens with it. Oh, that one's not too bad. Yeah, so just try to stack. Problem is we can't really see.
Hold there.
A
We're
little
getting
more.
close.
We need more.
Let me see it.
Pretty good.
It's pretty
About at
close.
the bottom
It's
line.
pretty close.
That's right there. Yeah.
Right, but it needs to kind of stay in the spot and be at the surface. It can't drop down.
Yeah, I don't know what's going to happen when I let go.
Right, I know. It's kind of an experiment, I suppose.
I suppose.
Let's see. Well, so let's see if it stays.
Roger.
Yep. Be nice not to turn it too much. Gosh, that's not a good sign. But maybe it's fine. I don't know.
Looks all right.
Have you let go?
No.
Oh, okay.
Okay. Letting go.
Yeah. Okay. And once we get it injected, it does. I mean, I would prefer that they stay vertical, but you know, if they tip over, they tip over. But so now here's a question. Ideally, we would inject them all at the very end after they're lined up in a row, but I understand that's difficult. And so if we need to inject it now, we can.
Either way.
I think for timing, maybe it's better because we're pressed for timing. We should probably just inject it now.
Roger.
But we keep track that this was the first
Can
one.
you
It's going to
tilt
be
up on
exposed to
bubble
more
and
lights.
show me the squeezy? I need another vantage point on it. Beauty. Thank you.
And so for this one, since we're going into capillary tubing, I think we want to do this kind of slow and steady. Don't do a big jab kind of thing, if possible.
Slow squeeze, is that what you're saying?
Yeah.
Okay, here we go.
I'm not sure where to look.
Yeah, it's
Yeah,
We're
pretty
looking in
perfectly
the,
right behind.
The camera there in front of Michael is a 90-degree out view.
yeah, but I can't see the end of the tubing.
Can we
Yeah,
zoom
we'd have to reposition for that. Okay, that's a full squeeze.
I can try
Yeah,
a quick.
so I mean, if it's squeezed, then it should be fine. It should have gone in. It's only 1.25 milliliters.
So only one squeeze?
Yeah, I mean, if you squeeze it more than 2 milliliters, we should be golden. But you know, I didn't see anything.
I can choke up on the jaw a little. It'll get, you know, that squeezed it with 4 fingers as opposed to where it's flat on the jaw there.
I think, you know, 2 milliliters is not very much. So I'm just thinking it probably went in.
Okay. You want to show me the azimuth shoulder of the manipulator, please? I could zoom back out too.
Data, are you just giving those a serial number as they're not really samples yet?
Yeah, so those are going to have the number now so that we're logging the current sensor data. So that is 45.
45?
Yeah, and then we will not relog them when we recover.
Okay.
Yeah.
Because now, once it's in the seafloor, that's its own little environment. And so it doesn't really matter exactly what is going on when we come back.
Hey Dan, if we're going to pack these, I wonder if we want to do one further away and then bring it closer so we don't chew up a lot of lateral left to right real estate.
Yeah. Wishing I had the parallel jaws now. I'm going to have to do a funky grab on this one too.
So hold on a second. Why don't you do the ones you can reach easier, because we're not going to get all 5 of these in if we have to leave the seafloor in 20 minutes, 15 minutes.
I'm already on this one, so I'm going to
Okay.
go with it.
Dan's going to get you there. Will take us down to a delta of 0. And then we'll start coming up. That'll buy us
I
another
can actually
40
start
seconds.
coming up on the winch before the ROV
Yeah.
lifts off the
Yeah, that's
seabed.
what I'm saying.
Oh, remember so they're all configured effectively the same way, so you could rotate it
Rotating.
a little more. So about in that orientation, I'm not sure if we can see it, but it's probably
That should be good there.
the
Okay,
best likelihood anyway.
here we go.
Okay.
Yes, you want to go
Happy
further?
there?
You want to go further
Sure.
away if you're going to stack
No,
them?
I can't do that. Sorry, I
Copy
gotta go
that.
left or
No
right.
worries.
Arc.
Maybe
Deja
this one will
vu.
be perfect. I guess not. Oh, not bad.
That was pretty good.
Just try to auger in and then pull. Yeah, but then we need a hole for it.
I think it's filling in underneath when I
Okay,
pick it up. So
I see. Maybe a
Hold
hair
on just a
more,
sec.
if
Hair
you
more.
can. Perfect. There we go.
Yeah.
Yeah, let's try to release and then do a squeeze if it stays in place, which I'm thinking it should.
I was trying to.
Yep.
That's all right.
Sorry. What you see is what you get.
It's okay.
Understood.
I was trying to pan the camera left so he could zoom in more, but
I can get a little more. Why don't you set up for your squeeze and I'll go in.
I mean, the core looks good. It's stable.
You
Yeah,
can try
it
some
would be
zoom
nice
there.
if we could
Yep.
zoom a little bit. Yeah.
There.
When I pan the camera left, there you can see something.
Okay. I don't see the end of the.
The
But
end
that's
is-
okay. Just
do you want to see the
just
top?
go for it. No, I don't need the top.
Oh,
I'm
I
looking
know
for
the
that
white.
little,
I can.
the end of the capillary
Show me
tube
a
that's
bubble, Mike,
in.
please.
Say again?
Bubble on that thing.
Yeah.
I need the 90 out. There you go. Thank you.
Yeah, no, I can't see anything.
Try and get it in the bottom of the jaws this time.
So you're squeezing?
I am squeezing.
Okay.
That one definitely got a better squeeze.
Okay.
Happy?
I think so. I didn't see anything, but it's like impossible. Could just release. Yep.
Next
And
one.
it definitely squeezed it.
Okay. Mark that one in.
Okay, shoulder elbow please
1832.
on bubble.
Say again?
Shoulder elbow on bubble. We're gonna move a little faster here, so.
That was 046.
Roger.
Yeah, I like that, keeping it in that hole and just kind of.
Yeah, it seemed to work
Yeah,
all right.
well, it worked for that one anyway.
Okay, so Taylor Ann, I'm going to draw a picture on the back of this sheet of the orientation of the cores so we know what they look like when we go back.
Okay, can chase the arm around a little with bubble. Show me the end effecting.
We might be able to pull a video freeze as we lift off when we're done to show the alignment as well.
Okay.
Reorient. Very good.
Okay, zoomy zoom.
Yeah, more. That one we might have to go to another spot. I'm not sure. We need more. More.
One more.
Yeah, one more, please. Can't quite see. Oh, let's let it clear a hair. Gosh. Yeah, a little more. That's interesting.
I'd say that's it.
Yeah, stop.
Stop it.
Yeah. The mat, I don't know what's going on, but the mat kind of rolled off to the one side. That was interesting. Oh, just leave it in the hole, so to speak.
Okay. I'll zoom out just a bit in. That one's going to be a little more tippy. I'm not quite in as far.
You can push it down. Just go ahead and pick it up and kind of trying to decide. We're not going to clean this out, but so I think, okay, don't push it in too much further.
Yeah,
because it's going to over penetrate. So we're just trying to get it stable. It's up high enough so we don't, as long as it's
going to tip.
Okay, we can just inject it. Oh, but it's going to get covered up.
Yeah, I think the reaming.
That one didn't work so well.
Or it's just soft in that spot.
Okay. Why don't we
clean it out?
Leave that one there for now and try to get the other two, covered. I guess. You know, we might want to inject that one. Let's see how the other two work. But now, of course, we've kind of messed up the area. We don't have time to relocate.
Some bubble action there. End effector. Okay, I can see my shadow now. I know where I am.
Oh.
Can't get there from here. Might be able to get here.
Wherever you can get it.
I'll save that spot for the last one. How about here?
Wherever you need to.
Kind of rolled over, didn't it?
Yeah.
That's not good.
Try and get tricky here.
It's clean enough. Oh, I see.
That'll give me a little more reach. Clean enough?
Yeah.
Zoom in a bit there.
Yeah, it's clean.
Okay, happy here?
Sure.
There we go. Oh, maybe I can get here now. Save that spot for the last one.
I don't know. Going in fast might not be so, I don't know. We'll see.
I rolled a bit.
Sure. I don't know if you want to try to stack it.
Yeah, can do.
It seems like we're in the soupier stuff again.
Does.
Gotta be careful because they're
Yeah.
starting to affect the other ones.
Yeah.
Yeah,
Not liking?
not really.
Shake
yeah,
it out?
Yeah, shake that out.
Roger.
Okay, so like this is mission critical. So, I mean, at this point, I'm okay to just stay down for another 4 hours. If we have to get this done, we have to get it done. We'll just wait till the next watch change. This is that critical.
Okay,
So,
stand by. I'll go let Nicole know.
okay, thank you.
Mike will be happy with that.
All he'll have to do is 3 push cores, and he won't even have to go in the quivers. I mean, it's a little bit of a waste of time, but it's that important.
But it's important, then it's not a waste.
What time is it?
Well, they want us off the seafloor in 5 minutes.
It's 11:40.
Ah. Come out, Z.
The hard part is all that wasted material or unused material is covering up what were potential sampling sites.
And we don't really want mud on top of the ones that are in place.
Can I steal bubble real quick?
Right,
Yeah.
because you still want that clear view of light.
Yeah, I'm trying to do this down current of the other ones, but pulling it out up there.
You're not supposed to bring crinkly. Oh, I thought it was food.
Okay, do what we need to do.
Okay. So let's just try to get this done as, I guess, as quick. I don't know if we're supposed to do it as quickly as possible or.
Do it to make yourself happy with the situation, so don't push it.
Okay. All right. So, do you?
Zoom.
Well, do you want to try here? Do we have to move a hair? I don't know. We can't afford to cover up the ones that are in the sediment. We want them devoid of mud on the top.
Yeah, I can move.
Just float down current.
Yeah, I mean, it has to be pretty close, I guess.
Do you want to pick up that failed one before we move?
Yeah, that'd be awesome if we could.
Yeah, take your time at this point. Just let's get it right.
Okay. So,
Yeah, let me just move.
okay.
Okay, well, I'll be able to reach the failed one.
Okay, roger
Just
that.
going to slide
Very
to the
good.
right a little, change my head.
Be
When I
careful.
had